Watch me spiral gracefully, so elegant.
Next, beta sheet!
Precise, parallel, perfection.
And now, turn!
I flip the whole structure in just 4 steps.
Last but not least, look!
I may look messy, but I hold it all together. And I bind things.
Never underestimate the flexible ones. Loops and turns steal the show.
Good morning, everyone. Today, I'll be walking you through an essential but often overlooked part of protein structure: loops and turns. These structural features play critical roles in folding, flexibility, and function. And today, we will explore exactly how. By the end of this session, you should be able to first, identify different types of protein turns. Second, use visualization tools to label loops and turns. Third, differentiate loops and turns from A helices and beta sheets. 4. Predict turn regions from amino acid sequences. 5th, explain their role in protein folding and function. And finally, contribute to analyzing what happens when protein structures are disrupted. Before we dive into turns and loops, let's refresh on the main types of secondary structures: the A helix and beta sheet. These are stabilized by hydrogen bonds and form the backbone of protein architecture. Now, can you identify them in these diagrams? Loops are longer, flexible, and irregular. They often set on the surface of proteins and connect elements like helices and sheets. Turns, on the other hand, are shorter, just 3 to 5 amino acids long, and reverse the direction of the polypeptide chain. They may seem small, but they are vital to the compact 3D structure of proteins. To summarize, turns versus loop. Loops are longer, surface-faced exposed, and variable in shape, while turns, on the other hand, is short, directional reversals, stabilized folding. Together, they enable proteins to fold correctly, position active sites, and interact with other molecules. Classification of protein turns. Let's now classify. We have beta turns, the most common, and Y turns, which is tighter and least frequent. Next is pi turns. They are broader and rare. Each of these differs in length, hydrogen bonding, and tightness of the band they introduced. Beta turn types. Now, there are 4 main types of beta turns. Type 1. Common, involves a small 4 residue fold. Next is the type 2, which is tighter, usually requires glycine at position 3. Now, type 1 and type 3. They mirror images of the first two, with opposite backbone angles. These subtle variations allow protein to fold in very specific ways. Y turns and pi turns. Y turns use just 3 residues and form a very tight loop with a hydrogen bond between residues and I+2. Next is the pi turns. In contrast, it utilizes 6 residues, are looser, and usually occur within pi helices. These are less common but still functionally important, especially in flexible or regulatory regions of proteins. Amino acids that favor turns. Certain amino acids are turn-friendly, like glycine. It is small and flexible, perfect for tight spaces. Proline is a rigid and creates natural bends, often initiating a turn. When we see these residues in a sequence data, we can sometimes predict where a turn might occur. Why loops and turns matter? Why care about these structures? Mainly because they enable compact folding, connect functional domains, and are frequently found at active sites and ligand-binding regions. And they are hotspots for mutations that cause diseases. Let's take a real-world example, the CFTR protein, which stands for Cystic Fibrosis Transmembrane Conductance Regulator. This protein forms a channel that transports chloride ions in and out of cells in places like the lungs and pancreas. When the CFTR gene is mutated, the protein it encodes can be made incorrectly, misfolded, or degraded before it even reaches the cell membrane. This leads to impaired chloride transport, which causes the buildup of thick, sticky mucus, the hallmark of cystic fibrosis. To explore loops and turns in real proteins, we can use molecular visualization tools like PyMol or Chimera. With this, you can label loop regions, identify specific turns, and predict how mutations might affect them. To review, here's a structure. Can you identify a turn, a loop? Which type of turn is likely here based on the residue spacing? To wrap up, loops and turns are not just connectors. They are essential to protein shape and function. They are often involved in active sites, binding, and mutation. Understanding their types and roles give us deeper insight into protein biology. Any final questions? Thank you for listening, and I encourage you to try identifying loops and turns in your own molecular models.